Let's now discuss how bacteria are classified. Now, why do we need to know the classifications? So practically, so that they are easier to identify. And the faster we are able to identify them, the faster we are able to treat a patient who is infected. So this now refers to the diagnostic part of microbiology. So to be able to successfully diagnose the bacteria and the infection it causes, there are three terms stated in your textbook. So identification, classification, and nomenclature. Identification is actually the process by which the microorganism's key features are delineated. Once those features have been established, the profile is compared with those of other previously characterized microbes. So we need to be able to isolate the microbe and then distinguish it from other microbes by noting the features of the isolated microbe through different criteria, which we will be talking about in a while. Once the microbe is identified, it can then be assigned to the most appropriate taxa or classification. And then, it can be given the appropriate genus and species names or nomenclature. Giving the microbe a name is important because it allows medical professionals to communicate with each other, especially regarding management of the diseases caused by each particular microorganism. Now, what are the ways we can identify bacteria with? We can actually make use of two general ways. Number one, by looking at their genotypic characteristics or how they are genetically made up, and also by looking at their phenotypic characteristics or the observable characteristics. So examples of characteristics used to identify bacteria include first by looking at their macroscopic morphology on culture. So that this means how they grow on artificial media as observed when you look at them when inspected with the unaided eye. Media used in the culture of bacteria are either selective or non-selective. Okay? So if the media used is non-selective, this will support the growth of many different bacteria. For example, blood agar and chocolate agar. We can also use selective media if we want to eliminate or reduce the large numbers of irrelevant bacteria, considering that certain bacteria only grow on certain media, or that some media will not allow certain bacteria to grow. For example, media with bile salts allow gram-negative enteric bacterial growth, but inhibit gram-negative mucosal bacteria and most gram-positive bacteria, like McConkey agar, for example. It contains bile, and it allows the growth of gram-negative rods only. Or media with colistin and nalidixic acid, such as CNA, colistin and nalidixic acid, CNA, blood agar, which selects for gram-positive cocci only. We also have differential media. When we culture certain bacteria, some of them produce characteristic pigments or colors, and others show zones of clearing around colonies grown in the presence of insoluble substrates. For example, using McConkey agar, pathogenic Salmonella and Shigella that do not ferment lactose form white colonies, while others that do ferment lactose, like E. coli, form red or pink colonies. So this way, we are able to know what bacteria are growing on the culture. Now, when the bacteria successfully grow, we can then identify the characteristics like the size, the texture, and pigmentation of the bacterial colonies, even the odor. So here are some characteristic colonies of some bacteria. Mycoplasma appears like fried egg. Bacillus anthracis looks like a medusa head. Pseudomonas has odor like grapes. Anaerobes are usually foul-smelling. Bacteria with capsules and slimes show a mucoid appearance. For example, the Streptococcus and the Klebsiella pneumoniae. When colonies appear smooth, it usually represents growth of bacteria with surface antigen and polysaccharides like Salmonella. When they appear rough, they are usually bacteria that are devoid of capsules and surface antigens and are usually avirulent, except Mycobacterium tuberculosis and Bacillus anthracis. So, of course, tuberculosis and anthracis, these are very virulent bacteria. Okay, so now we've tried to look at them macroscopically. Another way to identify bacteria is to actually look at how they look like under the microscope. And we will be doing this during our laboratory sessions. We will be taking note of their shape, arrangement of cells, intracellular inclusions, cellular appendages, and of course, their color after staining. Now, this is usually done more often in diagnostic microbiology than culturing a specimen because culturing bacteria takes longer. It takes a long time for the bacteria to grow. And staining and looking at them microscopically is usually faster to do. And once identified, you can immediately proceed with the specific management of the infection, such as the use of specific antibiotics known best for the identified infectious agent. Okay, so now one by one. We start with shape. How do we classify bacteria according to shape? Okay, so some bacteria are spherical. They are called cocci, coccus if singular. If they look like rods, they are called bacilli. There are also others that are oddly shaped, no? So spiral or corkscrew in shape, the spirochetes, for example. These bacteria can take on different kinds of arrangements depending on their activity. So if one coccus sits beside another coccus that he considers his best friend, they are called diplococci. For example, Neisseria, gonorrhea are diplococci. If they group together in force, they are called tetrads. For example, the Gafkia tetragena, which causes soft tissue infections. And then some may also group together to form a cube-like formation of 8 cocci, or the Sarcina formation. For example, Sarcina lutea, which causes GI disease. Now take note that using light microscopy, you will only usually be able to see structures in two-dimensional form or 2D and not 3D. So take note that Sarcina in 2D may look like tetrads on light microscopy, okay? So do not expect to see cubes of 8 using a simple light microscope. Okay, now some may form chains of cocci, and we call them streptococci. Of course, some may also just cluster together like a bunch of grapes. We call them staphylococci. Bacilli can also take on similar arrangements, no? Mycobacterium tuberculosis are bacilli that appear as diplobacilli. They go in pairs. Streptobacilli, or rods in chain arrangement, like Bacillus subtilis. Alright, so bacteria can also be identified by their structural appendages, or motility organelles. So some bacteria have flagella, which are thread-like appendages made up of flagellins, which are organs of locomotion that are attached to the cell membrane. We can further note differences in the arrangement of flagella. They can be monotrichous or polar, like that of Vibrio cholerae. They can be amphitrichous, or one on each pole, or lophotrichous, having multiple polar flagella like the Pseudomonas. And also peritrichous, meaning the flagella are distributed over the entire cell, okay, like Salmonella and Escherichia. Another kind of appendage is the pili, or fimbriae. These are rigid surface appendages of many gram-negative bacteria, which are shorter and finer than flagella. So these pili are composed of pilins, and they have adhesins at the tips for attachment. There are actually two kinds of fimbriae or pili. The first one, the ordinary pili, are for attachment to host cells. So these are actually responsible for the virulence of some bacteria. And then the second kind is the sex pili, for attachment of donor and recipient cells in bacterial conjugation. Okay, so you also have to know the endospores. So during conditions when nutrients are scarce, some bacterial genera are able to form spores. Essentially, the cell transforms from an actively metabolic and growing state to a dormant or resting state, with a decrease in cytosol and a concomitant increase in the thickness and strength of the cell envelope. The endospore remains in a dormant state until favorable conditions for growth are again encountered. So this is actually a survival tactic, no, by certain bacteria to survive extreme conditions. Endospores can be located differently in different bacteria, and you can actually use them to identify due to these characteristics. For example, Bacillus subtilis have central endospores, and Clostridium tetani have terminal endospores. Okay. Some cells also have capsules. A capsule is a structure immediately exterior to the peptidoglycan layer of gram-positive bacteria, and in gram-negative bacteria, exterior to the outer membrane. And this is made up of high molecular weight polysaccharides. Actually, they do not function as an effective permeability barrier, or doesn't even add strength to the cell envelope, but it serves to protect the bacteria from attack by our immune system.